Hi, my name is Ganesh, and I'm a junior in Phoenix, Arizona, interested in biomedical engineering and medical science.
Hi, my name is Ashwin. I'm also a junior, and I'm interested in the application of AI to medicine.
And together, we created Sculpt, our project for AzSEF 2025. So the dorsolateral prefrontal cortex plays a fundamental role in executive decision-making, social cognition, and other functions of the brain. But scientists today, we don't know much about it. Yet, this same area of the brain affects many neurodegenerative and neurological diseases such as schizophrenia, autism, and major depressive disorders, which affect millions globally. So, because we don't know much about this area of the brain, things like targeted drug therapies, radiotherapies, etc., we can't do it because where are we going to aim it? How are we going to do it if we don't know much about this area of the brain? So we created Sculpt, a multi-omics deep learning framework that can resolve this issue with computational power, a multi-omics framework, as well as spatial and temporal recognition of the brain.
So we used two data sets for data extraction. So the first one we used was for the spatial component of the project, and we used CNN for that, and we used the cell X gene data set for that, and then we used the CNN. And then for the RNN, we used the MindLab data set, and we tracked the gene expression of the neurons from a neonatal to a 39-year-old. And through combining both of these data sets, we could detect biomarkers associated with neurodevelopmental disorders and neurodegenerative disorders as well.
Exactly. So the problem we have is that mapping amino acid localization and gene expression in the DLF PSC, it's incredibly hard, and existing methods like integration of spatial, temporal, and multi-omics data. So the question we have is, how can a spatio-temporal multi-omics deep learning framework improve prediction of amino acid localization and gene expression in the DLF PSC? So our claim is that our model, Sculpt, as we integrate all these CNNs and RNNs and analyze these multi-omic data sets, it can actually improve this prediction accuracy by looking at the spatio-temporal aspect of these data sets. And we do this with statistical analysis and on par with the CNN using UMAP, PCA, and GMMs. So just another insight, the main programming language you use to build this model is Python. We use libraries from TensorFlow, Matplotlib, and PyTorch. And the model architectures are used with CNNs for spatial analysis and RNNs for temporal analysis that we talked about before. And MindLab was used for temporal gene expression, and CellX gene was used for spatial data sets. And so this is the problem framing in essence. So you have all this multi-omics data that we got from the data sets here, MindLab and CellX gene, and we fed this multi-omics data through an encoder and decoder. So the encoder, this is what we call a PCA, and we'll talk about that more later. But we apply this to each of the kernels through a Gaussian kernel. And what it does, it downsamples the image, and that's in part with the CNN, and that allows it, that's what we call the encoder step, which downsamples it. And this is called GMM, Gaussian mixture model, which upsamples the image back and brings it back from 1D to 2D to 3D. And we'll talk more about this later in statistical analysis part of this presentation. And this obviously results in telling us where it's going to be in the spatial part, because obviously it's a CNN, so we used it for the spatial aspect of this project. So this is the model building and multi-omics framework that we use. So we use two computers, our personal machine, which is a MacBook M3 Pro chip with 18 gigabytes of RAM and memory. And this took about 5 to 6 minutes of processing time for the CNN, obviously, because we ran it for 10 epochs, and it ran for approximately 30 seconds each, 34 seconds each for each epoch. And so when we ran this on a lab computer at one of our university labs, it had the specs for that was a 96-core CPU, an AMD Threadripper, and 96 gigabytes of RAM with two 6000 ADAs, and ADA NVIDIA GPUs. So for this, it took approximately 30 seconds total, so about 3 to 4 seconds per epoch, which was a crazy increase, and that was pretty interesting to see. And this means it was an 88% decrease in processing and training time. And so this is the model building. We started out with 14 layers, and the biggest change we had was changing it from Con 2D to Con 3D, and all 2D layers to 3D, which allowed us to have an increase of accuracy from 80% to 92%, which was really interesting to see. And so, yeah, this is what I was talking about before with the encoder-decoder steps. So this would be PCAs, this is GMMs.
Okay, so what was the first part of this project? So we basically made an accuracy predictor for the amino acid sequences we had. We got these amino acid sequences from the UniProt IDs we acquired from each gene, and we basically made a repository of those. And so how this amino acid sequence accuracy predictor works is that we enter an amino acid sequence, and then it basically gives us a percentage of how much it matches with our already given data set. And so you can see right there, we entered an example amino acid sequence, and then it gave a top match with locality-based accuracy, which was PHB2. And you can see the PHB2 gene over there. And then as we ran the accuracy predictor over and over again, the loss decreased each time. So the predicted accuracy was getting better each time we ran an epoch. And so what were the results? So the mean accuracy was 92%, highest at epoch 5 for both the CNN and RNN. And then the R-squared value, which is basically the mean error, was 0.87. So that was really good. And we also used the cross-validation 5-fold split, which is when we train 4 parts of the data and we test 1 part. So this is basically used to prevent overfitting and also make the machine learn better. And we got an F1 score predictive accuracy of 0.91 and an error of 0.015 across each part of the data. And then the error matrix also revealed a strong prediction accuracy for all the neuron subtypes. And then the neuron maps also revealed distinct amino acid expression gradients. So basically, each part of the DLPFC expressed a unique amino acid expressed unique amino acid acids. And then we also benchmarked against the Allen Atlas brain repository, which allowed us to get a 94% accordance with it. And then also the RNN or the temporal models revealed an increase in glutamate expression, which was mostly found in broad mammals at 8, 9, and 46. And so we also used PCA, or principal component analysis, and this was used for dimensionality reduction, and it basically filters out all the noise and redundancy while also keeping the variance. So it just keeps the important parts of the actual data. And so we use the equation x prime is equal to xw, where w is the eigenvector matrix, and this one just saves all the data in the direction of maximum variance. So we want to get all this parts of the data that are important. We want to make the machine learn from the low accuracy parts of the data, not just the high accuracy, just to prevent overfitting and all that. And so we also use the Gaussian kernel because we have to. It's a CNN, so this is nonlinear data. So this basically works with the PCA in reducing the dimensionality.
So next I'm going to talk about the Gaussian mixture model. So the Gaussian mixture model, as I talked about before, was the upsampling part of the data. So it turns it from a 1D image to a 3D. So it goes from 1D to 2D to 3D, right? Because PCA brings us from 3D to 2D to 1D. This will do the opposite. This allows us to identify spatial patterns and distinct amino acid gradients within the DLF-PFC when we actually train the data and see what's actually where the amino acids actually are within the this area of the brain. So beyond fundamental research, Sculpt has clinical applications, right? Because you can have personalized medicine applications, which will help with diseases such as Alzheimer's, Parkinson's, other neurological and neurodegenerative diseases. So it helps in that aspect. And in accordance with the $500 billion Stargate AI infrastructure project, which will help personalized medicine treatment, this would actually fit in very, very neatly with that. Because of its implications as to the accuracy predictor, where we can enter an amino acid sequence and see where it is in the brain when it's going to be expressed. And essentially, all this does is tell us how and where and when amino acids and genes get expressed, and that's really important for drug deliveries, drug therapy, targeted therapies, which will help with these neurological and degenerative diseases. That's all we have. We're excited. Thank you so much.
Thank you.